Right, morning everybody. So my name's John, and I'm going to talk about DNA methylation signatures and their role in identifying metabolic disease for the purposes of this talk. And it's really interesting to hear an update on how DNA methylation signatures are used for the diagnosis of cancer. Fantastic. But what about the opportunities for using DNA methylation for the prediction, early diagnosis, and better risk stratification of other chronic diseases? The chronic disease that I'm interested in is primarily interested in, my group primarily interested in, is type 2 diabetes, which is the trillion-dollar disease paradigm. Diabetes is a lifelong condition. It doesn't kill you the way cancer kills you, but you have to live your life with it. So there's a tremendous amount of morbidity, as well as ongoing healthcare expenditure for affected individuals. It's a really common condition. It affects 1 in 11 adults globally. That means there are presently about 450 million people living with diabetes, needing regular drug treatment day in, day out. And within the next 10 to 15 years, that number's predicted to rise to about 650 million people living with type 2 diabetes daily. It's a disease that's not fun to live with. You've got the treatment implications, the cost implications, but as you progress through your life with diabetes, you're at high risk of developing complications such as peripheral neuropathy, retinopathy with blindness, cataract, chronic cognitive impairment, kidney disease, heart attack, stroke, and even an increased risk of cancer. So as people progress through the disease, they accumulate additional comorbidities that are, in turn, a great challenge for them. Diabetes is a growing problem. It's increasing in burden in all regions of the world. And the greatest increase in burden is predicted to be in the South and Southeast Asian region, with an approximately 80% increase over the next 20 years. So it's a big global problem. It's also a specific problem for Singapore, which is why Singapore declared a war on diabetes about a decade back. What's the problem? Well, there are multiple challenges. One is that the burden of diabetes, like other parts of the world, is rising rapidly, with a prediction of a 30% increase by the year 2030. Secondly, there is a specific susceptibility to diabetes in Asian populations that remains unexplained, and that's illustrated by the middle panel, showing the incidence over 10 years of diabetes across the life course amongst European men and women, and the incidence of diabetes amongst Asian men and women, showing that at every age in life, Asian people are unfortunately at about 3- to 4-fold higher risk of developing diabetes. And after decades of research, we still have no idea why. On the right-hand side, we can show convincingly that it's not, as one illustration of the data, that it's not driven by something simple like adiposity. So if we take, for example, the burden of diabetes by adiposity amongst Europeans, this is the burden of adiposity. This burden of diabetes broken down by adiposity in Asian people. So if you want to say what is the equivalent metabolic health between Asians and Europeans, a lean South Asian individual has the metabolic health of an obese European with a BMI 10 points higher. This is BMI adiposity doesn't explain the health inequity. And if we look within Singapore, we see that there are tremendous differences also, not just between Asians and Europeans, but actually between Asian subgroups. Here we've got and it's not just in diabetes, it's in a range of related metabolic disturbances. So here we've got age-related plots for the burden of diabetes and related metabolic problems in the ethnic subgroups of Singapore, blue for South Asian, green for Southeast Asian Malays predominantly, and red for East Asian, predominantly Chinese. And we see that there are tremendous disparities in glucose levels and levels of insulin sensitivity, HDL cholesterol and triglycerides as markers of the insulin-resistant state, as well as with visceral adiposity. What's driving this? We currently don't know. Where's the next slide? There we go. There's the next slide. Okay. So It's a big problem, and it's a big problem for Singapore. It's a big problem for Asian populations. Now, a little bit more background. Diabetes is the endpoint of many years' progression from normal. So most of us start out life with normal glucose metabolism. Many of us will end up with diabetes, about 1 in 10. Over the lifetime, about 20 to 30% of us will end up with diabetes. And as you progress from normoglycemia through to diabetes, you go through a phase that's called pre-diabetes. And that's where we start to see early abnormalities in glucose and insulin signaling and glucose metabolism that can be detected through measurement of raised glucose, the primary marker of glucose metabolism, or through measurement of glycosylated hemoglobin, the traditional long-standing measure of long-term glucose stability and control in human physiology. So there's a long track record of using pre-diabetes to identify people who are going to go on to diabetes. And there are multiple clinical trials that have evaluated how can we use pre-diabetes, glucose and HbA1c, to predict who's going to develop diabetes and bring them into intervention programs. It's great. It's got some strengths, but it's also got some key weaknesses as an approach. The problem with pre-diabetes. First of all, by the time you develop pre-diabetes, you've already got dysglycemia. And with that comes a significant increased risk of cardiovascular disease and of macrovascular cardiovascular disease, myocardial infarction, stroke, but also a significant increased risk of microvascular disease, including retinopathy, neuropathy, and nephropathy. So by the time you're pre-diabetic, you're already in a risk state. There's also, once you're pre-diabetic, you're the bloke who's about to fall off the cliff. It's very late in the progression from normal to disease. And if you look at the progression rates to diabetes, amongst Europeans, half of them will progress to diabetes over 10 years, people with pre-diabetes. And amongst Asian populations, the data show it's as many as 80% progressing to diabetes within 10 years. Once you've got pre-diabetes, your future trajectory is pretty clearly mapped out. In the next decade, you're going to progress to diabetes. And then we've got the problem that although there are some promoted interventions, traditionally lifestyle intervention, weight loss, physical activity, or metformin treatment, those are the classic therapies established by the Diabetes Prevention Programme and other studies, this shows the progression to diabetes amongst people randomized either to placebo, blue, metformin, red, or lifestyle intervention, purple. And yes, it's true that metformin and lifestyle intervention do lower the risk curves. But by the time you're like 10 years out, is that a material difference in outcomes? The risk is reduced from 50% to 40% with aggressive lifestyle intervention maintained over 10 years. So what we're saying is that the available interventions are only modestly effective. What do we need to deal with this? Diagnosing prediabetes is too late. We need to get to people's risk when they're still normoglycemic so we can give earlier intervention and avoid this high-risk phase. We also need better interventions that target the underlying biology better so that we can have therapies that are more effective. So this is the program of work that I try to pursue. And my approach is using the population-based strategy. There are other wet lab, animal, whatever type experiments, but my living laboratory is the population. I map out my team, my group, me and my colleagues. We map out the genetic and environmental and behavioral choices of large numbers of individuals as to create a population cohort. Within the population cohort, we go on to identify the subset of individuals who develop disease. We use that data set to carry out molecular phenotyping, identify biomarkers for disease, understand the relevant systems biology and molecular pathways, generate molecular insights, and from those molecular insights, translate the insights into precision medicine through better biomarker discovery and through policy and action, including drug discovery. So the population cohort bit is pretty straightforward. You collect 100,000 people with dense phenotyping and biological samples, and I'm going to talk a little bit about the molecular phenotyping that we do to advance biomarker discovery and precision medicine therapeutic insights. Okay, molecular phenotyping. It's a big bucket. It's multi-omic research, huh? So we're familiar with genomics, genetic variation as underlying DNA sequence variation, genomic regulation, epigenomics on top, leading to transcriptional variation, proteomic variation, and then metabolic and lipidomic variation. All of these layers can be informative, no doubt about it. But for the purposes, my own research enthusiasm and my group's trajectory is focused upon DNA methylation as a disease pathway and as a biomarker paradigm. I hope most in the audience know pretty well what DNA methylation is about, but as my sort of simple guide, DNA methylation I perceive as being the light switch on the genome that either turns genes on or turns them off, with typically high methylation, hypermethylation being the trigger for gene silencing and chromatin condensation. It's a bit simplistic because the reverse can also happen, that gene activation can lead to changes in DNA methylation, but that's the sort of my simplistic epidemiological view on things. Why is methylation a potential goldmine for discovery? First of all, DNA methylation is well established now to play a critical role in cellular programming, that is the development from the ovum to the complex organism, as well as programming of cell lineages and tissue structure and function. But it's all, we all have the same DNA sequence, but we develop into this fantastically complex multicellular organism. It's also involved in adaptive responses to environmental and behavioural stressors. I like it because it represents a site that proximally integrates genetic exposures, environmental and exposures, and behavioural exposures. It is the landscape that controls the genome and so determines, as an upstream process, and determines downstream consequences. I love the tremendous molecular heterogeneity. If we do metabolomics, I send my sample off to Metabolon, and they send me back 1,000 metabolites measured using state-of-the-art mass spec. But there are 1,000. That's quite a lot. But in the methylomic space, I have 30 million CpG sites to unravel. It's tremendously complex information, with complex information the opportunity for better discoveries. And there's substantial evidence now implicating disturbances in methylation as being key drivers of chronic disease etiology. And I'd highlight this beautiful paper that was in Cell last month, demonstrating how epigenetic entropy and the loss of epigenetic control that happens as we become more chaotic in life, as chaotic as time passes, increasing entropy, may be a key driver of senescence, dedifferentiation, or they call it X-differentiation of cell lines, and the aging process and the development of chronic disease. So the epigenome seems to be like a great place to start for biomarker discovery and understanding pathways. All right, now the last bit before I get into the sort of what we're doing now is to fess up and say, we've been looking at DNA methylation for some time. And this is a study, this was one of the first large-scale studies. These paired studies were one of the first large-scale studies of DNA methylation variation in relation to non-cancer, non-communicable diseases globally. The work is almost a decade old now, but it sets the scene for why the next round of experiments. And what we did back in the day was we did a simple nested case control study of instant diabetes. So we took 2,500 people, all of whom were well at baseline. Half of them went on to develop diabetes, the other half didn't. And we carried out methylation profiling of whole blood using what was then state-of-the-art, the Illumina 450K methylation array. And we were really tremendously excited to identify 5 molecular loci, methylation loci, that predicted risk of future diabetes. Just 5, not a large number. But the effect sizes are quite encouraging. Whereas we're used to become accustomed to common genetic variants that confer risks of now 1.05 to 1.1, even the strongest SNP for type 2 diabetes, TCF7L2, only carries a risk of 1.5 to 1.7. So here we're getting risks of 2.5, something if you're in the top quartile compared to the bottom quartile for one of these molecular markers. So, potentially quite discriminatory. And if we club the methylation marks together, we can get odds ratio, and we show a nice interaction with obesity. That it's additive risk on top of traditional risk factors such as obesity. So, compared to normal people with normal weight people with lowest levels of methylation, if you're obese and have got raised methylation levels, you're at 10-fold higher risk. So we're seeing interaction between the two that gives additive information. Okay, so that's good, a good step. But we also noted this phenomenon, and this is that the DNA methylation arrays, whilst they were state of the art at the time, we thought, well, is this really enough information? So we took the trouble to resequence one of the candidate loci to see if we could get more information out of it. And this is the top locus. It's the TIC-SNP gene. It's a regional plot of TIC-SNP locus. And each of these numeric codes represents one of the CPGs at the TIC-SNP locus. And the only one measured by the 450K methylation array is this one. Just this one across the 100 or so present at this site. So whilst the methyl array is it's state of the art for 2015, it actually at the time captures less than 2% of epigenomic variation. So how can we then go forward into a biomarker program saying, we think we've got biomarkers for diabetes prediction when we're only measuring 1% of the methylome? And how can you persuade people to do functional studies? How can you even conceive what the molecular mechanism is when you're only got one signal across the whole genetic interval? What's the likely mechanism, and what's the appropriate experimental design? So building on this work, over the last 5 years, our program has set out with 3 key goals. First of all, it's to improve our understanding of how methylation markers predict diabetes through a combination of methylation array studies involving larger sample size, but also through a combination of both whole methylome and targeted methylome sequencing. Then to carry out functional genomic evaluation so we can get some insights into molecular pathways, and then thirdly to determine potential predictive utility for diabetes prediction. Taking those 3 study aims in turn. First of all, improving our understanding of biomarkers and methylation markers that predict diabetes. And for this, we've now scaled it up, and scaled it up. It's still tiny in comparison to GWAS literature. We're now studying 6,500 individuals, of whom half develop future diabetes. So we've increased our sample size 3-fold. We're also scaled up by now using the Illumina methyl epic array, which has got 850,000 markers as opposed to 450,000 markers on the 450K array. So a 3-fold increase in sample size and doubling of the coverage of the epigenome. And what do we get from this? Well, we get tremendously more discovery. And specifically, we go from having 5 genetic loci methylation markers linked to diabetes now to 420 loci predicting future diabetes. 314 discrete loci, and this shows the Manhattan plot. It's all over the genome. The top associations are highly plausible for being involved in metabolic regulation. We see ABCG1, SHREB, TIC-SNP, HANAC, and UFM1, all of whom have got good, compelling, preliminary preclinical data linking them to insulin signaling and or risk of metabolic disease. So let's now do, so that's great. So we've really pushed forward the number of methylation loci linked to future risk. I'm going to touch on how we're exploring those into potential biological pathways. I'm going to come back to the resequencing thing in a little bit, but I'm going to jump into the biological pathways. And the question we want to ask is, are any of the methylation markers involved in disease etiology? There might simply be a confounding effect, a side effect of the underlying exposure and not involved in etiology at all. And we can address that question using genetic markers as causal, as anchors for Mendelian randomization studies, that is, genetic instruments to assess causality. And the basic principle of the MR experiment goes, here's the disease we're interested in, here's the CPG site that's predicting future diabetes, let's work backwards and find some genetic variants that influence those methylation marks. If the methylation is causally driving diabetes, then a SNP that drives methylation will also be directly driving type 2 diabetes. This is a Mendelian randomization experiment. And if we do this up at scale, which we can do, we've got 400 loci to pursue, and we've now got large data sets, which we didn't have 10 years ago, to describe methylation QTLs and large data sets that describe the genetic variation underlying diabetes. We can now identify at least 4 genetic loci that show a causal link mediated through methylation linking that show 4 methylation loci for which genetic evidence supports a causal mechanism, causal pathway linking the methylation disturbance through to type 2 diabetes. Potential opportunities now for function, for in vitro and in vivo studies to examine the causal pathways involved. And are these plausible? I'll just what does this colocalization look like? Is it convincing? Well, here is the BAIP2, which is also known as insulin receptor substrate P53, just to show that it's known to be involved in insulin signaling. And on the left-hand side, we've got the GWAS association p-value. On the right-hand side, the SNP methylation association. So SNP methylation is orange dots. GWAS signal for diabetes is blue dots, and what we see is that there is colocalization with the top SNPs for both the GWAS signal and the methylation signal intersecting with each other, providing evidence of a shared underlying genetic architecture. I've highlighted that these are strongly plausible candidates that are crying out for some functional studies. Jared2, for example, is involved in development of the endocrine pancreas, and the conditional ablation of Jared2 in mice impairs endocrine cell differentiation and generation of beta cell differentiation programs. Whereas BAI-AP2 is also known as the insulin receptor substrate. It's involved in insulin signaling, particularly in the brain. Okay, right, so we're identifying some good candidates. How else are we identifying candidates? We're also looking to see, well, how are these methylation markers potentially linked to gene expression signatures, and we can do that both in cis and in trans. And to do that, we've carried out RNA sequencing of 1,200 of our samples. 10 years back, there were no RNA-seq data sets in Asian populations in which to unravel these signatures. So we've had to create these data sets as unique repositories. So we've also gone to the trouble to generate RNA sequencing profiles of Asian populations as a unique global resource, and we can then test the methylation marks for their relationship with cis gene expression. We find that 65 of the genes are linked to 65 of the methylation marks are linked to cis gene expression. And again, we can use genetic instruments to assess whether those relationships are likely to be causal. So we can start to pin down causal pathways linking methylation to gene expression and also methylation to type 2 diabetes. Well, we can now start to bring this multi-layered data. We've got sequence variation, we've got methylation levels, we've got gene expression cis and trans, we've got disease phenotypes in trans, and we can layer on top of that protein-protein interactions and gene expression covariation patterns, and we can now start to build up plausible biological pathways that link exposures through to disease outcomes for testing in vitro and experimental models. Okay, So where are the results of these in vitro and experimental studies? Why haven't we done them yet? It's because I need to talk about this bit next, which is I need to get through this bit before I come back to fine-tuning molecular mechanisms. So I've shown that we've identified 400 markers. I've shown that they look plausible biologically and we can create coherent pathways. What I'd also like to now share with you is how we can use the information for disease prediction. And we're familiar with the utility of molecular information for prediction of disease using, for example, polygenic risk scores, in which we use the sum of the effect sizes across the identified loci to allocate scores, genomic scores, to people with disease or without, and we see some separation of the cases and controls that allows us then to categorize people as being high polygenic risk, intermediate polygenic risk, or low polygenic risk. So within our validation data set, not a discovery data set, we've done the discovery, now we've gone on and validated, we've now constructed methylation risk scores to complement polygenic risk scores. And these are the distributions of polygenic risk scores, left, and methylation risk scores on the right, in our cases blue and our controls red. And we see that polygenic risk scores are, yes, they're shifted a little bit towards the right, a little bit of a right shift, but the methylation scores are much more right shifted compared to the controls. And that translates into substantially better discrimination of our diabetes cases from our controls. Future diabetes, not talking about prevalent, we're talking about people who are going to get diabetes in the future. On the left-hand side, and in this bar, I've got risk of future diabetes associated with quarter 4, quarter 3, or quarter 2 of polygenic risk score compared to quartile 1. And so if you're in the highest quartile of polygenic risk, your risk of future diabetes is doubled. Okay, so here's a room full of 50-odd people, 15 of you or so have got a high polygenic risk, and I can say that you're about twice as high at risk compared to people in the lowest quartile over there on the left. It's it's helping, but it's not really, it's not really cutting the cheese. So let's look at methylation risk score, and again we've got a quartile plot. Now if you're in the top quartile, you're now at 7-fold higher risk compared to quartile 1. If you're in the second quartile 3, 3.5-fold higher risk, and even quartile 2 is achieving a 2-fold higher risk, substantially better than polygenic risk. So now, my 60 people in the room, 15 people over here, I can tell you you're at 7-fold higher risk based upon this compared to 15 people in the lowest quartile. That's quite concerning to hear that you're almost 10-fold higher risk compared with people on the left-hand side, just in a normal, random group of people. Now, within this, we can drill down a little bit further, because I've said that we already identify people as being at risk based upon type 2 diabetes and based upon HbA1c and glucose. So I want to show conventional measures. What I argued was we need to go upstream. So now I'm repeating the risk estimates in people who are strictly normoglycemic at entry. They have normal HbA1c, normal glucose by the strictest international criteria. And I apologise that my research fellows now did this plot as a risk per 1 standard deviation as opposed to per quartile, and that leads to confusion. But what we're showing now is risk from polygenic risk, blue, per 1 standard deviation of risk score, risk from methylation risk score from 1 standard deviation in risk score amongst normoglycemic individuals. And this is amongst all normoglycemic individuals, and this is amongst people who've got obesity, showing this interaction again that the combination of adiposity and raised methylation risk score is particularly troubling. Now, what is a risk of, say, 2 per 1 SD mean in real terms? Well, think about your normal curve. If you go from, say, quartile 75 to quartile 25, that's about 3 standard deviations from left to right. 3 standard deviations from left to right is 2 times 2 times 2, 3 lots of standard deviations, 2 to the power 3, which is an 8-fold difference in risk between one end of the spectrum compared to the other. So this is providing discrimination even within normoglycemic individuals. Okay, now I also set out the challenge that we don't understand and we can't well predict the ethnic differences in risk within the Singapore population. So this is now showing methylation risk scores by ethnicity. And this is the plot to look at. We have got the blue Chinese as the reference, who are the lowest risk group in Singapore, and we've got the South Asians and the Malays in red and green respectively, who are the high-risk populations in Singapore, and the methylation risk scores are shifted approximately one standard deviation to the right. That's equivalent in terms of risk to, for example, systolic blood pressure being 10 points higher or cholesterol being 1 millimole per litre higher. This is a tremendous increase in risk captured by methylation risk score. Okay, so where's the clinical translation of this? Seems like it's compelling. But we have to go back to this problem, which is our final challenge to resolve, which is that what we've measured, these apparently quite discriminatory risk functions and these apparent relationships to biology, are based upon measurement of single CPGs at what are complex genetic loci. And if we're going to really understand how we can use this for precision and understanding biology, we need to get a better handle on the locus and not just on individual CPGs. So that has led us to say, well, let's try to use resequencing approaches to understand what's going on at the loci. And we have adopted a whole methylome approach to sequencing in this experiment, doing whole genome bisulfite sequencing in 250 people with type 2 diabetes and 250 controls, 500 whole methylomes, amongst the largest experiments of whole genome bisulfite sequencing done globally. We used an Illumina platform, a Swift Biosciences library prep kit, and we did 150 base paired end sequencing to a target depth of 30x. We did this about 2 years ago, and it cost us almost 2,000 Sing dollars per sample. So this was $1 million experiment, even with only 30x sequencing. And what do we get out of this? Well, I'm going to show you I'll show you what we get out of this. This is the association across the gene. First of all, we did an experiment. Let's just first of all see if we can get any additional loci through this whole genome bisulfite sequencing experiment. And we were disappointed, but not greatly surprised, to find if we take a whole genome view in these 250 cases, 250 controls, there were no additional loci that were standing out as being associated with diabetes. But also, we didn't find the loci identified by a methylation array, which seems a bit concerning, doesn't it? But what we have to recognize is the profound limitation of a 30x whole methylome, because whereas the microarray is highly reproducible with a minimum average error of 1.4%, 30x sequencing has a mean average error of 7.5%. The total variance in experiments determined by the biological and technical errors, and when the technical error is high, that reduces the power of the experiment. So if you're going to use next-generation sequencing for methylation loci, for refining methylation loci, you need to be aiming for 100x sequencing at least to give precision, 100x depth in your methylation calls. And that is not achievable with current pricing with a whole methylome approach. What have we used? So we expected that. That wasn't a surprise. What we actually wanted to use the whole methylome data was to map out which of the other markers at the locus might be interesting. So here is now one of our exemplar loci, SERPINE1. Here's the key methylation marker identified, and what we see is a nice clear tower of local correlation. This looks like the biological interval that might be driving the disease. This is where the correlation lies. And within our preliminary data set, we can say, well, here's the marker that's measured. How do these other markers link to future diabetes? And how does methylation across the locus link to diabetes? This is the methylation marker measured by the array. These are the other methylation markers at locus. And actually, some of them look to be doing better. So this provides the robust evidence on which to justify resequencing across all 400 loci. How are we going to define the interval to resequence? Well, we've done it by doing a systematic evaluation of the pairwise correlation of CPGs in either side of our sentinel CPG site. And the interval of correlation is narrow. It's only 500 base pairs either side. How are we going to map these out to high depth? Well, this is where it was fantastic to meet the Twist team, who told us about their targeted approach to Twist methylation sequencing. And of course, it comes with a glossy, exceptional performance, high sensitivity, design flexibility, gentle enzymatic conversion. But all credit to Twist. They stepped up to the plate and put their money where their mouth is and did a fantastic pilot study with us to show proof of principle. And what we've done is a pilot study using the Twist Methylome panel, 5 million markers, 16 samples split equally between 2 labs, 1 set run in Singapore, 1 set run in the United States, so completely blinded, done in duplicate, and both sets run with a 500x sequencing, attempt to deliver 500x sequencing. What do we get for that? We get 70%, at least 70% of sites measured with 100x coverage. We get 6.5 million CPGs measured on this panel. And most importantly for me, we get high precision across these sites with mean absolute errors of less than 2%, comparable to microarray. So this enables us to go forward robustly and now take 2,000 samples, do 500x sequencing using a targeted sequencing panel that includes all the markers at the relevant loci with results expected in the next 3 months. And this will tell us now what are the best predictive biomarkers for DNA methylation profiling. It'll enable us to leverage our prediction from a 7-fold risk between quartiles to hopefully something even stronger, 10, 15, who knows. But it'll also give us confidence about where the locus-related methylation disturbances are to inform well-considered, well-designed experimental evaluation of underlying genetic mechanisms. I've overrun. Thank you very much for your attention. I hope you've enjoyed some of the insights.